We're on first line. We're on the last bit of innate immunity. So let's see. I think we're going on to activation of complement, right? So we're actually going to start out with a video. And before I play this one even, let me make sure I got all my things on.
Complement.
Okay, we do.
It consists of a complement.
All right. So, actually, I'll just pause it when I get to the point where I want to talk. So, let's do that.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function.
Right, so C stands for complement, and then there's the number associated with that protein. And then some of them break into fragments, remember? So we have things like C3A. Which one of these does C3A participate in? Inflammation, cell lysis, or opsonization? C3A, which one? Inflammation? Inflammation, right? It affects the membrane, it affects the permeability of the blood vessels, right? And that's part of the process of inflammation. C3B, is it part of inflammation, cell lysis, or opsonization? Opsonization. What was the clue there? Remember C3B. Think of binding, right? Because opsonization is basically tagging for destruction, right? C3B binds to the surface of the cell and tags it for destruction. So what's the other one for inflammation? C3-5A. Right? And that's a chemoattractant for what white blood cells? Who do we want to come when we have inflammation that are phagocytic? Neutrophils. What do we call that complex that causes cell lysis? It's abbreviated MAC, but it's important that you know what MAC stands for. Membrane attack complex. So it's a bunch of these proteins, right, that form a donut-shaped hole in membranes. So who is more susceptible to complement lysis, right, by the MAC complex? Gram-positive or gram-negative? Negative. Megan's got it. Why, Megan? Yes. Because the gram-negatives have that outer membrane, and again, this complex inserts into membranes. What would it have to travel through first for a gram-negative to get to the membrane? The cell wall, right? And that's why it's harder, right, and less likely to be damaged by this complex. So this is a cascade of events, right? So almost like dominoes. Once it starts, right, then it's going to keep going. And you're going to get, hopefully, if C3b can bind, it will bind to the cells and tag them for destruction. They're going to opsonize them, right? These substances, C3a and C5a, are going to help with the inflammation, that influx of neutrophils to help with the phagocytosis, which is going to be enhanced by opsonization, right, of C3b. And then, even more so, we could directly destroy the cells with the MAC complex if it can insert into the membrane of those cells, which is more likely to happen with gram-negatives. So that's the important end to this story, right, is that we can directly kill them with the MAC complex. We can infiltrate the area with neutrophils and hopefully eliminate invaders. We can enhance this ability of our phagocytic cells by opsonization. And then again, the three molecules we need to know for these processes are C3A and C5A for inflammation And C3b is the actual protein that is the opsonin, right, that tags for destruction. I don't make you guys memorize all the proteins that are involved in the MAC complex, right? Just know that it's a membrane attack complex and that it is a series of those proteins that form a donut-shaped hole. So now in this video, the rest of it, we're going to talk about how do we start this process. So this is really great, right? We get opsonization, we get cell lysis maybe, we get inflammation, the influx of neutrophils to kill. How does it start though, right? And so that's what the next thing that we're going to look at, right, is how does it start? And again, we're going to look at the start, right? And they're going to go through the steps in the middle, and it's going to be a generalization. It's actually more detailed than this, but you guys do not have to know the in-between details, right? You have to know how does it start and what does it end, right? You don't have to memorize the middle, right? But pay attention.
The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation, they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5A and C5B. C5A enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components, including C6, C7, C8, and C9, to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
So the middle is scary, huh? Aren't you glad you don't have to memorize that, right? So let's summarize it, though, because there's some important steps in there. And one of the activations they don't talk about. So it's a series of proteins, right? These proteins are circulating in your blood and fluids in the inactive form. Right? They've got to be activated for this cascade of events to progress. It augments activities of the adaptive immune response. So anybody know what component of the adaptive immune response was seen here? It activated the classical pathway. What binds to antigens that we produce? Antibodies, right? So notice in the classical pathway was antibodies that started this reaction. So we've got to activate, once they become activated, right, this series of reactions happened. The three ways that activation can happen, two were shown in this video, the alternative pathway and the classical. The third is the lectin pathway. Now the names, unfortunately, are somewhat confusing sometimes. The classical pathway, just let me switch to this slide right here. So the classical pathway, what does that mean to you when someone says classical? Original, old, right? And you could kind of say this was, because this was probably the first way that was discovered. Because again, antibodies were very readily, easily studied in the beginning. They're proteins that our body produce. So they were probably one of the first products of the immune system that were studied. So this was probably the first pathway discovered, right? That antibodies could activate complement. The alternative pathway was, of course, discovered as an alternative as our studies became more detailed. In the truth of the matter, most of the time, if it's the first time you've been exposed to a particular antigen, if the complement cascade is being activated, it's via the alternative pathway, not the classical. Because if it's the first time you've seen an antigen, do you have antibodies against it? No. It takes about 7 to 14 days to make an antibody upon first exposure to that antigen. So if this is the first time, chances are you're not activating complement via the classical pathway because you don't have antibodies against that pathogen. Instead, you're doing the alternative. C3b and C3a are constitutively made, right? We're always making them. But they've got to be activated for it to progress further. And one of the ways that activation can happen is when the C3bs bind to the surface of the microbial invaders, right? Then we get that progression to that common stage for all of these pathways of creating an enzymatic form of C3 that continues to split other C3s into C3a and C3b, right? It just keeps doing it. Yeah, okay. So Bridget asked the question, she goes, when you go get a flu vaccine, they're like, you're not going to be protected right away. It's going to take about 7 to 14 days for your immune system to mount the response that's going to protect you, right? So the minute you get that shot, you're not protected, right? But you're bettering your chances because 7 to 14 days later, hopefully your immune system makes a response, makes antibodies. So if you're exposed to that virus, then those antibodies will bind to it and neutralize it and help stop the infection sooner. And antibodies is the next thing we're going to talk about. So the other pathway that wasn't shown in the video, and I'm going to switch back to my other slide where it has the description for you guys. This pathway, the activation requires mandan-binding lectins. So it's a pattern recognition molecule, right? It's recognizing a particular pattern. So just like before, we recognize lipopolysaccharide and peptidoglycan and flagellin protein. Mannan, which is a sugar, it's a polymer of the sugar mannose, right? So a whole bunch of mannose sugars hooked together to form a molecule we know as mannan. It's found in microbial cells, right? We don't make mannan, microbes do. And again, it's a common substance, a common molecular pattern for microorganisms. So it makes sense that we would have the ability to recognize it. And in this case, we're not just going to recognize it, we can activate complement when we recognize it. But the same thing it's going to happen for any of these activations, whether it's antibodies via the classical, C3b binding via the alternative, or the lectin binding recognizing the mannan polymer. We're going to converge at this point, right? That's common for all of these guys. That's important to remember. is that we're going to get this continual breakdown of C3 into C3A and C3B. And remember, our C3B is our opsonizer, right? It's going to bind to the microbial cell walls or membranes and tag it for destruction by our phagocytic cells. Our phagocytic cells have receptors for C3b. They readily will gobble up any microbe coated in C3b. Remember, as I said, I always remember C3b, B for binding, right? This literally binds to the surface of the microbes and tags them for destruction. That is known as opsonization. C3a, right, is important in the inflammatory response, getting those phagocytic cells there so they can destroy the cells that have been opsonized. So then the next major step in the process is, again, breaking C5 into its two fragments. C5a, which again is involved in that inflammatory response, specifically as a chemoattractant, says to neutrophils, come here, come here, come here, right here, right here, we need you right here. And then C5b goes on to the initiation of the formation of that MAC complex. But again, a membrane attack complex, so it's only going to insert into membranes and therefore cause cell lysis by that complex inserting. It's a pretty cool system though, huh? Right? It can kill, it can tag for destruction, it can get the cells that we need there, right? It's a really good system. But it's relatively generalized, right? It's not detecting any specific bacteria, unless, of course, it was activated by antibodies. In that case, we're having a specific interaction. But in essence, it's a general reaction, right, to foreign substances that's always there in our body, always on the ready. These are some of the proteins that we talked about that leak into the area too during inflammation, right? When those blood vessels become permeable. So, how is it that we don't destroy our own cells? Well, C3b won't activate when it's attached to our cells. We have regulatory proteins that stop C3b that binds to the surface of our cells from activating complement. Microbes, of course, they don't have protection against this. So the C3b binds, right? Other complement proteins bind, as we saw in the animation, and we get activation of complement. For our cells, regulatory proteins stop C3b that's bound from interacting with the complement proteins. So like when we talked about phagocytosis, can some bacteria avoid phagocytosis? Yeah, some of them can even escape out of the phagosome, right? Or are not even destroyed by the phagolysosome. So do you think some of them can stop C3b from binding? Yeah. Some of them even do that by coating themselves in our own regulatory proteins. They have receptors that bind to our regulatory proteins, which stop C3b from binding and activating complement. Yeah, tricky bugs. This is why we still get sick and still have disease, right? If this was a perfect system and nothing could get past the system, then we would never get sick, right? That would be wonderful. But it's not the case, right? And so whenever we have illness or disease, it's because it's evaded our immune system or even used our own system against us. And in this case, they can coat themselves in our own regulatory proteins and stop C3b from binding and stop it from being activated complement system. So the last thing that we're going to look at before fever is interferon response. And this is a really good animation on that. So I'm going to go ahead and play that one for you.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So interferon, as it implies, it interferes, right? In this case, with virus replication. So as you know, viruses have to get inside of our cells in order to use our ribosomes, right, to make their proteins. And they use our building blocks to do so. We have receptors. Remember, we have receptors for double-stranded RNA inside of our cells. And for viral RNA in general, and viral components. So when a virus goes in, and if it triggers those receptors, it's going to trigger a particular response, in this case, an interferon response. Right? So RIG-like receptors. So these are in the cytoplasm, right? These are going to detect that presence. Interferon is produced by that cell that detected the invasion. But the unfortunate thing is they can't respond quick enough to save themselves. But instead, what they do is send out a signal to neighboring cells. That signal is in a chemical form we know as interferon. Right? So that signal gets sent to neighboring cells to basically warn them, right? It's screaming, run, right? Or in this case, prepare to fight, right? The bad guy is here. You need to prepare to fight. How are they going to prepare? They're going to produce enzymes that will destroy messenger RNA. The problem is, in this case, they're not going to really be able to stop the destruction to themselves in the process. So again, these cells are going to basically, if the virus gets in, it's going to trigger that system to activate these enzymes they've made because they've been warned. And they're going to prepare to fight, and that is that when that virus gets in, they're going to activate those enzymes, and those enzymes are going to destroy the virus, but they're also going to destroy the cell itself. It's one of the unfortunate side effects of this. The good news, though, for the organism, for our body, right, is that a few cells have sacrificed themselves to stop the replication cycle of the virus, right? If it can't replicate inside that cell, then it can't go and infect other cells, right? We stop that process. Unfortunately, oh, I know what it is. It's me. I made the mistake of leaving my email open. I was like, it happened earlier this morning too, and I was like, what is that noise? You forget what these alerts are sometimes. Okay, there we go. Fixed. Sorry, I apologize for that. Whew, crazy week. So those cells too go through apoptosis. What does that mean? Cell death. Cell death, but cell death without what? Without inflammation, right? Without inflammation. The resident macrophages that are there will clean up the mess. We don't need to call in neutrophils and more macrophages to take care of this problem in this case. But we definitely need, right, the cells to stop this cycle. And the unfortunate thing is that obviously some cells are still going to be eliminated in the process, but the good news is the virus is stopped. So you'll sometimes hear commercials on TV nowadays for the different drugs that are out there for different illnesses and diseases. They'll talk about how it doesn't have interferon, right? And so we have actually, you know, outside the body manufactured interferon to help with some viral infections. So for some viral infections to boost and help your body, they will give you interferon so that, again, your cells will get prepared, and so that way if the virus does enter the cell, they'll destroy it and stop the cycle. Make sense? Right? It's all about stopping that viral cycle. And the good news is we're multicellular organisms, right? We can afford to lose a few cells, right? We just don't want this to keep going, right? We don't want them to keep multiplying and keep infecting ourselves. So that's how we defend ourselves against viruses, with interferon. The last for our innate responses, these generalized responses that we have, is fever. So what happens when we have fever? So that's a signal to increase core body temperature. Why is increased temperature beneficial? Heat, yeah. Why is heat beneficial? What do we use heat? We use heat to kill microorganisms, right? That's why we cook our foods, right? We can use heat to kill. So if we get hot enough, we could potentially kill the microorganisms that are invading us, right, if we're too hot. The other thing is that chemical reactions in general happen faster with heat, right? So heating up even ourselves a little bit is going to help us move faster, right? Our cells to do things faster. So it's a twofold thing, right? We could potentially kill microbes by getting too hot for them, and we can respond faster because with heat, chemical reactions happen faster. Fever also is usually a pretty strong indicator of bacterial infection. Not always. But when you have a fever that's not going away and is actually increasing pretty steadily, it's usually a bacterial infection. And the reason for this is that, unfortunately, some bacteria out there produce cryogens. Cryogens are fever-inducing substances. We produce our own cytokines that are classified as cryogens. These are referred to as endogenous because they came from within, right? We made them. We told our own body, get hot. The unfortunate thing is that some microbes produce substances that are cryogenic as well, which will cause us to increase our core body temperature. We're not telling us, the bacteria is telling us, get hot. So this is an exogenous. It comes from outside of the body. Well, the problem is that when you heat up, you sweat, right? And then you get convection and you get cold, and that's when you get chills, right? So you go through, depending on the cycle of the fever, right? Your periods where you're really hot, then you sweat, and then you'll be chilly, and then, the cycle goes over and over again, depending on the duration and what's going on. So the problem is that the microbes themselves sometimes are making us get hot. We are not controlling it, right? When we don't have control, this is bad, right? And this is why sometimes fever can be bad, because sometimes we don't have control over it. We are not causing it to keep going. So this is when you end up in the emergency room at 10 o'clock at night with your kid because they've got an ear infection, and it's progressed so far that the infection is so bad and the bacteria themselves are causing the fever to continue. Right? This is not something you can easily see as a parent, and for some reason this always tends to happen at night, not during the day. Right? If you're a parent, you will probably experience this, or you'll be really glad if you never do. I unfortunately had to do that and went all the way to tubes in his ears because of the problems with little kids' ears and other problems that some children have. In my case, my poor son inherited my allergies. So it was an allergic type problem where fluid was building up in his ears and it wasn't draining out. Then you get stuff that goes up your nose, right, settles in your inner ear, and you get an infection. The good news with that, though, is usually antibiotics will knock it out. The bad news is, like for my son, if you have allergies and you continually have this buildup and you're not stopping the allergic reaction, then you're going to keep getting infections, right, until you can stop the cause, until you can get at the problem. Little kids are also more prone to ear infections because their ear canals, their stationary tubes, are more lateral. As your head grows, they pitch more. So it's easier for the fluid to drain out of your inner ear back into your nose, and less likely for stuff to go up from your nose and stay stuck in the middle ear, in the inner ear. It's an unfortunate part of our anatomy, right, that changes over time. So in that case, right, you've got this fever, it just keeps going, right? And chances are, like I said, it's bacterial, because you wouldn't do that to yourself, right? Because what happens if we get too hot, right, and it just keeps going? We're going to kill ourselves, right? You can only get so hot for our cells, enzymes, and proteins before we start destroying our own cells, right? And so that's the scary thing with that. And that's the thing what some people don't understand, though, is that, yeah, fever is a natural response, but also sometimes bacteria cause the fever, right? And they're in control, and we are not. The good news with bacteria, most of them we can kill them off with what? We can get antibiotics and kill them all. Right? But with some viruses, too, you get fever, right? Sometimes we get a fever response. And again, that's our body, right, signaling. And unfortunately, it's not really an effective response usually against viruses, except for the fact that we get a little bit of speed up in our system, right? We're going to do things faster. And that's probably why we still get a little bit of a fever response sometimes with viral infections, because we do have that increased speed up. But they're not growing, right? Viruses don't grow like bacteria grow. Heat isn't really going to affect them in the way that it would affect potentially a bacteria. But they are made of proteins too, so maybe some of those proteins are damaged by heat, but I don't think the level that we can get to would be effective. So that's it for Innate. So at this point, right, sometimes over the weekend or before class on Tuesday, I highly suggest, right, trying to do the Innate Connect assignment, because we finished it up. And now we're going to move on to adaptive, and we're going to start out with my favorite molecule, and